Hi students, once again I am meeting you to discuss about the applications of biotechnology. So far I studied about the applications of biotechnology in relation to what we have the various products, medicinal products, and secondly about the gene therapy. Now coming back to the third one application, that is nothing but stem cell therapy. This is the advanced technology, now it is being applied for various fields. Now what do you mean by the stem cells? They are present both in adult as well as in embryos. The stem cells are undifferentiated cells found in most other multicellular organisms. All the cells are of the same type. Only after differentiation they get actually converted into different cell types. So undifferentiated cells are called the stem cells, giving rise to new cells of different types in the body. Now they maintain their undifferentiated state even after undergoing numerous mitotic cell divisions. As in the case of, you see that when meristematic cells of plants, they are always dividing, producing more and more cells, later differentiated into permanent tissues. The same manner in the animals too. We have the cells. They are always undifferentiated, dividing by mitosis, producing more and more cells. After even, you see that when many mitotic divisions, they remain in the undifferentiated state. For example, the bone marrow cells. We have some stem cells, myeloid cells, monoblast cells, erythroblast cells, like that. Now, what is the importance of stem cells? Now, you see that various researches have been made, still going on, about the stem cells. So it has the potential, that the stem cell research has a potential to revolutionize the future of medicine, because we are depending on new technology. One such technology is stem cell therapy. When compared to gene therapy, it is less expensive, and that one is, see, the cost is high, but this one is less expensive. So stem cell research has the potential to revolutionize the future of medicine with the ability. What's the ability they have? Regenerating their diseased organs, or what we have, the defective organs. By means of stem cell technology, we can replace the diseased organs, either kidney or heart, etc., or the defective organs in the body. That's the main importance. And they have the capacity. They are capable of what we call self-renewal, renewing themselves. That is called the regeneration capacity, renewal, what is called rejuvenation also we can see. And also they have cellular potency. Cellular potency means they have the potential actually to generate, either we have different types of cells, or many types of cells, or only one type of cells, or we have a few types of cells. They have the potential. That is called cellular potency, having the ability to produce the different types of cells, or only one type of cell, or the entire organism. That is called cellular potency. So in the animal body, the stem cells differentiate into all the cell types, and these cells are derived from the three germinal layers: ectoderm, endoderm, and mesoderm. All these layers are derived only from the stem cells during embryogenesis. Also in the case of adult, they develop into what we call the different types of cells, as in the case of blood cells, etc. So there is another importance, generation of different types of cells, and these cells have been derived from the three primary germinal layers: ectoderm, endoderm, and mesoderm. They are formed during what's called embryogenesis. Then another importance, the most important and potential application of human stem cells is the generation of cells and tissues. And those generated cells could be used in cell-based therapies. Therapies, treatment, cell-based therapy, what we have, stem cell-based therapy. Even the human stem cells are used to test the new drugs. That is the next advantage or the next importance. I write down on the board. So the applications, the most important, that is what we have, and potential applications of human stem cells, that is the generation of new cells and tissues, and such cells and tissues could be used in cell-based therapies. Again, human stem cells are used mainly for, I mentioned, to test new drugs. These are all some of the importance of stem cells. I mentioned about the importance of stem cells. Of these importance, the most important and potential applications of human stem cells is, I mentioned about, the generation of cells and tissues. And those cells and tissues could be used in cell-based treatment or therapies. Again, we have the human stem cells that are also used to test new drugs. That's about what I discussed earlier. Now, what are the types of stem cells? There are two types of stem cells. One, embryonic stem cells found in the embryo, and another one, what we have, adult stem cells. So the embryonic stem cells are actually derived from, that one, that is the gastrula, or afterwards you have the blastocyst in the case of human development. We'll discuss. Now, what are the characteristics of embryonic stem cells? Number one, they are pluripotent. What is pluripotent? That is several. So these actually, the stem cells have the ability to differentiate into three primary layers. That condition is called pluripotency. So normally the stem cells are either totipotent or pluripotent or multipotent or oligopotent or unipotent. I'll discuss later. So pluripotency, the ability to produce the three germinal layers. That's called pluripotent. So I can produce the three primary germ layers, the ectoderm, endoderm and mesoderm. And that is called pluripotency, and the cells are called pluripotent. Formation or differentiation of the three germinal layers from the blastocyst or from the gastrula by means of, you see that one, morphogenetic movements called gastrulation process. That's about number one, property. Now we have to go for further other properties. Number two, the cells are multipotent and can differentiate into various types of cells in the body. That condition is called what is known as multipotency. Production of different types of cells, various types of cells which are related. For example, the blood cells. We have actually the stem cells of the blood can produce the lymphocytes, or we have any types of cells, the lymphocytes, the monocytes, or we have the neutrophils. Now all are related. In one way, they are doing what is called the function of the blood. And such a condition is called what is known as multipotent. Pluri means several, multi means many. So pluripotency refers to the formation of three germinal layers. Then multipotency refers to the formation of various cell types from these three germinal layers. And all these cells are related in their structure or in their function. One example, the blood stem cells can differentiate into what we have, the neutrophils, or lymphocytes and monocytes. That is called pluripotency as well as multipotency. I will discuss once again later. Okay. Now the second property of embryonic stem cells, that is what we have, these cells are multipotent. They are pluripotent, can give rise to different types of layers. What we have, the ectoderm, endoderm, mesoderm. The word multipotent refers to the formation of various cell types or the differentiation of various cell types from the three germinal layers. And those cells which are differentiated from the ectoderm, endoderm, and mesoderm are mostly related. For example, the mesodermal cells, the muscle cells, they are related. Or we have the blood cells, they are related. So that's a meaning for multipotency, production of various types of cells. So here I mentioned also I can differentiate into various types of cells in the body, but from the three germinal layers. Now the next property, where are they from? If we are taking the embryonic development of humans, after fertilization we have cleavage process. The cleavage results in the formation of what is called a blastocyst. In other animals we can call blastula, here it is called blastocyst, which consists of an outer layer of trophoblast, which is in contact with the uterine wall, and an inner embryonic cell mass. The cell mass later differentiated into epiblast, hypoblast and various structures. Now, the stem cells are derived from the outer epiblast. Okay, they are isolated from the epiplastic tissue. That's the one which later form the stem cells from the inner cell mass of blastocyst. the blastocyst, the blastula of human embryo during development, having an outer trophoblast layer in contact with the uterine wall, an inner cell mass, the embryonic cells, which later differentiate into different types of cells. Now here we have in later development the formation of outer layer called epiblast. That epiblast only giving rise to the stem cells. Then, when these stem cells are stimulated, they develop into more than 200 cell types in the adult body by stimulation, by activation. Then, so these cells are immortal, never die. Dividing once reach the maximum size, divide as in the case of amoeba, as in the case of what we call the meristematic cells. So they are immortal and can proliferate continuously and maintain their undifferentiated state even after many divisions. Okay, that's the nature of the embryonic stem cells. Now we have to proceed about the adult stem cells. Okay. Next one, adult stem cells. The adult stem cells are found in various tissues of children as well as adults. Each adult stem cell, also called by name somatic stem cell, can divide and produce or create another cell that is similar to it, create a cell which is similar to it. Then, if you are taking the nature of these adult stem cells, the adult stem cells are multipotent, can differentiate into various types of cells. So pluripotent, formation of 3 germ layers, multipotent, differentiation of various cell types from these layers. So multipotent and act as a repair system when there is a damage in a particular tissue because of injury. That place is being replaced by what we call that is these tissues. So they act as a repair system in replenishing, just actually rejuvenating what we have the tissues. Just actually what we can say rejuvenation of the tissues, actually just making the cells afresh. Human bone marrow is a rich source of adult what we call the stem cells. From there only we are getting the different types of blood cells. The bone marrow, particularly the red bone marrow, not the yellow bone marrow. The red bone marrow is concerned with what we call the stem cells for the production of different types of cells. Okay, that's about that one. We have to go for the nature or the property of what we call the stem cells, the different types, the different characters of stem cells. What is the nature of stem cells, or what are the properties of stem cells? The stem cells may be totipotent, or pluripotent, or multipotent, or oligopotent, or we have unipotent. So they exhibit their property in these ways. What is totipotency? The phenomenon. So totipotency is restricted only to a few cells in animals. The stem cells are up to, you see that one 8-cell stage in the embryo. But in the case of plant cells, all the adult cells, the leaf, stem, root, etc. are all stem cells acting as totipotency, exhibit totipotency, or they are totipotent. Any type of cell from any part of the plant body can give rise to the total organism. So if a single cell can give rise to the entire organism, then it's called totipotency. And the cell is called totipotent. For example, in animal cell, the zygote is a progenitor, it is a totipotent. Or the 8-cell stage, any blastobia can be separated and cultured to form a totipotent cell. But in the adult, you see that when we have the stem cells present, they are totipotent. That means a single cell can divide and produce the entire organism with all the different types of cells, and that's called totipotency total. One cell gave rise to the entire organism. Now the second one, pluripotency. Pluri means several. So it refers to the stem cells that can differentiate into the 3 germ layers. either ectoderm or endoderm or mesoderm. That property is called, that is pluripotency. Okay, the cells are called pluripotent. Give rise to only the 3 germ layers. The next one what we call multipotency. Multi means many. From any one of these germ layers, we have formed different types of cells or various types of cells. So it refers to the stem cell that differentiate into various types. And those various types of cells are interrelated or related. For example, the blood stem cell can give rise to the different types of what we have, the leukocytes, either the lymphocytes or the monocytes or the neutrophils. And that is a property of what is called multipotency. Many cells are formed of different types, related function from stem cells. Now the next one we have to go for oligopotency and then unipotency. Okay, let's continue. Now the next type or the next property, the nature. Oligopotency. The word oligo means a few. It refers to the stem cell that can differentiate into only a few cell types. For example, if you are taking the bone marrow, there we have the stem cells in the form of myeloid or lymphoid stem cells. They can differentiate into either lymphocytes or monocytes or neutrophils and not RBC. Only few cell types have been derived from that one, myeloid or lymphoid. That's why it's an example for oligopotency. Now the last one, unipotency. Uni means single. It refers to a stem cell that can differentiate into only one type of cell. So an example of erythroblast, the stem cells formed in the bone marrow, red bone marrow. It is differentiated into only what we call the RBCs, not any other cell types. So that is an example for what is called unipotency. Only one type is being differentiated from a stem cell. Example, erythroblast. That one gives rise to what we call the RBCs. That's about the nature or property of stem cells. Now in that stem cell therapy, the last part, we have what is called stem cell banks. What's the meaning for that one? A few words. Just like the blood bank for blood, the sperm bank for sperm, embryo storage, we also have the storage of stem cells in banks, what is called stem cell banks. Now what is the importance and what is called stem cell banking? Stem cell banking is nothing but the extraction of the stem cells, then processing of the stem cells and storage of stem cells for future use, for mainly actually for treatment for future use. That is when there is a requirement. When required in future, now these stem cells which are stored in banks are being taken and used. Now normally stem cells are used in banks. Stem cells are normally stored in banks, not used in banks. Stem cells are stored in banks. What's the main purpose of storing the stem cells? Stem cells are stored in banks specifically for use by the individual from whom the stem cells are collected, and the banking costs are paid by that individual. So every individual has a right to store their own stem cells in the stem cell bank. For that they have to pay the money. So the main purpose is actually the storing of stem cells specifically for use by the individual from whom the stem cells are collected, and what we have, the banking costs are paid by that individual for storage. For example, we can store the stem cell of umbilical cord for about 21 years at about a temperature of minus 196 degrees Celsius. Even after 21 years, you can take the stem cell and can produce an individual. But because of ethical issues, we are not producing any individual from the stem cells. This is because of the ethical issue. But it is possible to produce an individual from a stem cell that is collected from either umbilical cord or from the amniotic fluid, etc. So based on actually where the stem cells collected, we have the following types of stem cells banks. What are the types of stem cells banks? Number one, amniotic stem cell bank, and number two, cord blood banking. Here the cord refers to the umbilical cord, where we have collected the umbilical cord, stem cells, or the cells from the blood vessels. That's why it's called cord blood banking. Either the umbilical cord, the source, or the blood cells supplying the blood to the umbilical cord, the source of what is called the stem cells for banks. Now we'll discuss a few words about these two. What is amniotic stem cell bank and cord blood banking? Now the first type of stem cell bank, amniotic cell bank. Now it is a facility that stores the stem cells, those stem cells extracted from the amniotic fluid. See, the embryo is surrounded by or covered with amnion, an extra embryonic membrane, which is filled with what we have a fluid in which the embryo is floating. Now the amniotic fluid also contains a cells similar to the embryo body. So we can extract the cells from the amniotic fluid and store it in the amniotic bank, and that can be used for future purpose as stem cells. Now the next one, cord blood bank. Now the cord refers to the umbilical cord. Now the cord blood banking is nothing but the extraction of the stem cells from the umbilical cord at the time of birth. So during birth we are cutting what we call the umbilical cord. If we take a piece of umbilical cord and store it in minus 196 degrees Celsius, minus 196 degrees Celsius, it can be stored in the bank for nearly about 21 years. You can store it. After 21 years, you can take a stem cell from that one, you can produce the entire organism because it is a totipotent cell. So the stem cell can be stored in banks in minus 196 degrees Celsius. The stem cell from what we have the cord, umbilical cord, can be stored for about 21 years. Okay, now the cord blood bank is nothing but actually the collection of stem cells from the umbilical cord during birth. Not only the stem cells from the umbilical cord, the tissues, but also from the blood, which supplies blood to the umbilical cord. So, not only the umbilical cord and the cord blood. The cord blood refers to the blood vessels, cells, from where we are collecting what we have, the blood. So, this is an artery, a vein supplying blood to the umbilical cord, connecting the uterine wall of the mother and the fetus, through which only the supply of oxygen, the food materials all being transported, and also the waste materials transported towards the body of mother. So there is a connection between the fetus and the mother's uterine wall. So this is the one which contains stem cells, and now this is the fetus, and also we have the cord blood referring to the blood cells from the blood vessels of artery and vein of the umbilical cord collected. So not only you see that one, just we have the stem cells collected from the umbilical cord and cord blood, but also we have cells from the placenta, a vascular connection between the uterine wall of the mother and the fetus. So normally the umbilical cord arises from the placenta, present in the uterine wall. Amniotic sac, the sac covering the entire embryo. You see that one, this embryo is surrounded by amniotic sac. This is the amnion, and which contains amniotic fluid. Here only we have the cells we are collecting and storing for amniotic cell bank. Okay, now placenta, amniotic sac and the amniotic fluid are rich sources in terms of both quantity and quality. So these are the two major cell banks. We have in India different types. I am not telling about that one, not necessary in the syllabus. So that's about what we get the stem cell therapy. I want to conclude with a picture or a diagram how far the stem cells are differentiated into different cell types. A simple diagram is given in the book. Okay. So this diagram shows the undifferentiated stem cells. They are in a state of division, and then they are differentiated into different types of cells, either the nerve cell or the liver cell or the muscle cells or the red blood cells, or we have epithelial cells or any other cell types. Here I represent only a few as per the book. So many cells have been derived from what is called the stem cells. That's why it's being used in cell-based therapies. So that's all about what is the stem cell therapy. We have to proceed the next one, molecular diagnosis. We'll see in the next class.